Do do do. Hello, whoever's here. I'll try to start at 2 p.m. EST. I'm gonna get on my Twitter and tell people that I'm doing this. I kind of assumed for anyone who's here that I would start a couple minutes after 2. It is a bit strange being in here by myself and not being on a Zoom call, but you know, what are you gonna do? I feel like I should be like playing a video game since I'm on Twitch. I know that you're not a robot. Whoever you are, Santosh Manico. It's hard to tell, based on these usernames, who people are. But maybe it will remain a mystery. I'm currently testing. I think my cursor shows up on the screen. Great. And I'll wait until like 2:02 and 2:03 to go. So here just in a second. But for anyone that's here, I'm Wes Clawson. Dr. Clawson, I guess. It's weird. Yeah, new episode with me, Wes. Today I'm going to be talking about some of my work that I did when I was a PhD student. It will still influence some of the work that I'm doing now in Mike Levin's lab at Tufts, which I think everyone is familiar with. And for anyone who's here and is in the chat, I do have it very cleverly pulled up on my second monitor. So if I say something that you want to know more about, just say something in the chat. And I know there's a little bit of a delay, so If I'm like some of my slides are animated, like crappily animated, feel free to like mention what slide. I keep all the numbers at the bottom that should be visible. But I'm gonna go ahead and start because we're only supposed to do this for an hour, and I don't want to make sure that I get through what I want to talk about. So today's talk I titled Information Processing and Organization in Vivo and Altercations and Disease. And I've specifically put in vivo because Santosh mentioned that a lot of people here um, are doing stuff like in artificial neural networks. So here, I guess I'm coming from a more biological side. I got my PhD in neuroscience. So all the experiments that I'm showing today and some of the results that we're going to be looking at are all based in animals, so in rats. So let's get into it. Um, Thanks to Proteus for inviting me to do this. It should be pretty fun. Let's see. There we go. So my work, very similar to a lot of Mike's work, is based off of collectives. So collectives can be kind of whatever we think. So there's like things like bird collectives, right? So you have these interesting swarming behaviors. Ants can kind of exhibit collective behaviors in the way they build bridges, like from leaf to leaf. Um, Mike's lab does a lot of very interesting regeneration. So how to collect collections of cells after some kind of damage to a limb. How do they know that it's been damaged, and how does it regrow? And interestingly, when does it stop? Today I'm going to be talking about collectives of neurons, kind of like the collectives of us. Now we are more than just the brain, but today I'm going to be talking about the collective neurons in the brain. And speaking of collectives, this is the collective that helped me. The interesting thing about collectives is sometimes when they come together, they can show signs of intelligence, and we managed to do that just a little bit. So before I get any further, these are the people that helped me do this work. So my advisor for my PhD, Christophe Bernard, and then sometimes Demian Pataya, the wonderful Pascal Kilikini, and then some of the other PhDs and researchers that helped me along the way. But the story overall that I want to talk about is organization and how collective activity is organized. So when I think about um, organization, I think about the most organized thing in my mind is like a library, right? Everything, the books are in their place. You see, it's all very clean. It's even very quiet. And if you wanted to go through and categorize how this thing has been organized, it should be fairly simple. There's people who dedicate their lives studying to the best way to organize these things. So libraries to me are the most organized things I can think of. But I also like to think of the opposite. So the polar extreme to this would be something like this, which is a looted Amazon warehouse somewhere in Europe, I think. So you can see that there are still books. Like the content of the picture is somewhat the same, but it's all messy. If you needed to go in here and actually try to write a report on, the Renaissance, it would be very hard to figure out where to look for this. So this is the opposite of that. But what's really interesting is that there's something in between the library and this, and it's Christoph's desk, his office. And I think this is what a lot of our offices look like, or our homes sometimes. There's signs of both organization and of the kind of mess that we saw just then. So, there's shelves, there's stacks of these papers. Are they organized in some way? Maybe Christoph has a system. You see there's things in places that seem like they have a very specific purpose, and then other stuff looks kind of chaotic. But a lot of us find this kind of organized chaos the best system for us. So I'm going to be talking about how I think the brain is more like this than the library or the destroyed Amazon warehouse. So the data that I look at again comes from animals. So what we do is you record the electrical activity of the brain. So this top signal is called the local field potential, or LFP, and the bottom one are the spikes. So neurons in the brain fire action potentials. Collective action potentials produce electrical fields inside of the brain. So you can detect with an electrical probe both of these signals. And so that's what I'm going to be talking about today, and how this is organized. With the interesting question of, is this organization related to collective neural computation, which is a big question, which is why this slide is titled that way. Is the brain computing? It's a hard question, and one that sparks a lot of debate. But really what I'm going to ask is, if the brain is computing. There's going to take some kind of inputs, whether they be internal or external, do something with it, and there's going to be some kind of output, whether that be internal or external. And what I want to ask is, what can we say about how computation may be organized? And then if you have a pathological state, what does that organization look like then? Are there some things that are similar or different, or how does it change? And just to give a framework, I'm going to go over an example of a Turing machine, which I think maybe a lot of us are familiar with. So imagine this is a piece of tape that can have zeros or ones printed on it, and there's going to be a device that can read whether or not it's a zero or a one, and this tape can extend indefinitely in either direction. What's interesting is this head will follow a set of rules. So if it's in state A, you can see up here, and it reads a 0, it's going to follow some kind of action. Here it's going to print a 1, shift to the right, and then be in state beta. And that's what you see here. So it was a 0, it printed it into a 1, and shifted. And you can follow these rules over and over and over. In this specific example, there's a case where it halts, where whatever process it was running is now done and it stops. So this is kind of the computer classic theorized by Alan Turing, the Turing machine. And the reason I bring this up is it gives two really interesting signatures of computation, and that's state-dependent information processing and non-trivial state switching. So what I mean by the first point is that the things that it does are different depending on if it's in state alpha or state beta. So it processes the information that it reads, the zeros and ones, differently based on whether or not it's in alpha or beta. So it is state-based. And then there's non-trivial state switching, meaning that it's not just going back and forth, back and forth, back and forth. There's kind of a rule to when you go to one state or go to another. And these are kind of two classical signatures of computation. It's important to note that I don't think the brain is a Turing machine. This is just a model to get your thinking into what I'm going to be looking for are kind of these same signatures. And so what I'm going to do is we're actually going to talk about experiments with rats under anesthesia. Because if we want to look for something like state-based computation, what are some states? Like, what are the brain states? And that can be a lot of different things. But in anesthesia, in rat brains especially, but it happens in other mammals, when you're under anesthesia, your brain has these oscillations. So here, again, the electrical fields inside of your brain will oscillate at different frequencies. And it's what's shown here on the right. In this top graph, when you see the red, that means the power of a given frequency is very strong. And when you see something else like a lighter color, it means it's less powerful. So what we see is that the electrical activity in the brain is oscillating at around 1 to 2 hertz. And you can see it lasts a really long time. This is 1,000 seconds. That's a good chunk of time. And then you notice it kind of switches up into these higher frequencies, theta oscillations. It's like 7-ish. And it stays there really stably, and then it swaps back, and then again, and then again. And this happens we call these kind of global states because you can find them just about anywhere you stick your electrodes when you're under anesthesia. And these kind of mimic natural states of REM and non-REM when we have humans sleeping. So if I wanted to look at different computations that are based on states, anesthesia provides a good way to look at really long and stable states to see if there's something going on inside. And someone asked, do different species have different oscillations? Yes and no. So most oscillations are pretty general, like theta. We call that theta because what's interesting is across different species you can see similar patterns. Now, you should note that sometimes oscillations are spoken about a bit differently. If you're thinking about like human subjects to animal subjects, and sometimes the oscillation literature won't agree. Like there's a small thing here, a small thing here. But in general, most oscillations are somewhat similar across species, which is very interesting. So good question. And so what we're going to do is look at the spiking activity inside of these different states of slow oscillations and theta oscillations and talk about how they're maybe similar, how they're different. So if there's computations going on, maybe the neurons, the things doing the computing, let's check them out in these different states. And again, the big question being is organization of this stuff, of these neurons related to collective neural computation. where computation is the state-based information processing, like we saw in the Turing machine example, and then non-trivial state switching. But a really interesting question, and I'm gonna go through this kind of quickly, is if the brain is computing, what is it computing? I almost didn't include this, but I think it's interesting for the Proteus people. If you're computing, what are you computing? What is this computing? Mm, it's tough. And there's a really nice paper from Krakauer in 2017 that goes over these ideas of computation. And if you go back to the 1977, there are these guys, Mar and Poggio, who kind of posited computation having three levels. So the implementation, the what, the algorithm, the how, and then the computation itself, the why. So taken to a really simple example, if you have a bird, uh, and if you want to fly, that's the kind of the computation. That is the output you want at the end of all of this. Well, in fact, the feathers are the what of flying. You know, that's what gives the bird the lift and the ability to fly. But it's not just the feathers. If you have feathers, you also need the how. This algorithm, that's the flapping. So in this really simple example, the feathers are the what, the flapping is the how to get the flight. Now, here I'm going to talk about neurons in the brain maybe being the what, so they're kind of like the stuff of computation. However, the why is really hard to answer, and I would posit it's almost impossible to know. But we still could maybe investigate the algorithms. So if we think that the neurons are the feathers, how are they flapping? Um, So what we're going to do is how to investigate these kind of algorithms, the flapping. And again, we're looking for signatures of computation. One interesting thing to think about is what's the kind of the lowest level flapping that you could observe? Like, yeah, they're spiking, but maybe something a little bit more than that. And we think about the analogy to computer science. You can have high level languages like Python that can do a lot of very fancy things. But the deeper you dig into a computer, you have the hardware, right? The transistors that are actually flipping the zeros and the ones. But there's some kind of assembly language that helps make those flipping of zeros and ones and make them into something meaningful that you can do with at a higher level. So what would the assembly language of the brain be? So here we're going to posit that if you have a computing system, Claude Shannon theorized as well, or I mean Turing and Langton kind of theorized back in the day that kind of the most primitive operations that you can do in an information processing system is information storage, transfer, and modification. So for a system that's going to be computing, it needs to be able to hold things, storage. It needs to be able to move things around so that information can propagate throughout the system, and then it needs to modify it, right? It's kind of useless if all you can do is just move stuff around. I'm going to talk about 1 and 2 today, and the third will hopefully come in the future. Um, so again, we're going to talk about collective neurocomputing when we think about computing at the algorithmic level, and then I think that these kind of primitive information processes are kind of the assembly language of the brain that we can investigate. And then again, what I'm going to talk about at the end is what could a pathological system reveal about this organization. So epilepsy is my model system. The people that I worked with in the past worked on epilepsy, and so now I kind of do. And what's really interesting is there's in an epileptic brain, rodent, human, whatever, anything that has severe epilepsy, there's a lot of things that change. So you have changes to genes, you can have changes to cell signaling, to channels, to synapses, to networks. All of these things can be altered in epilepsy. But what's interesting is you will always kind of get these seizures that then back propagate to cause more changes. Things change, and at the end of the day, you're going to have these cognitive disorders. And what's crazy is you can have very similar cognitive disorders and very similar types of seizures, and what's underlying all of that, the thing that broke, can be very different. And this is kind of referred to as degeneracy. Is there's kind of many different ways for the brain to change, yet you get similar seizures, yet you get similar cognitive comorbidities like memory loss, for example. And often it is the case that we as scientists want to go in and say, okay, what changed? What channels changed that affect the computation? Something at a very, very low level broke, and now I'm getting seizures, which is not good. Like you're computing the wrong thing or something like that. And that can be really difficult, right? There's, okay, fine, the one small thing changed, or these two or three things changed, um, but it might be worth looking at how they're organizing their activity as well. Do the algorithms change as well? So kind of the main idea of my work thus far in my life and how I'm proceeding forward is, in control systems, sometimes the what can be very different in control to a disease state. So, like the what in control and the what in epilepsy, the neurons are different, the channels have been altered, the genes are switched. But how are the algorithms different? So that's kind of what I'm gonna, a summary slide of what I'm gonna talk about and how organization is related to all of this. I know it was a long introduction, but I wanted to make sure everyone like mentally was in the right space of how I think about these things and how I'm going to interpret them later. So there's kind of two projects that go through this stuff. One is information dynamics, and then the second is how these things change in epilepsy. And again, to go back through the experiments, what we do is you take animals, and they're of a certain age, you anesthetize them, and these are all acute recordings, meaning that you put the animal on some kind of recording platform. They're under anesthesia. They're kept very, very, very under anesthesia. You open up their head, you drill a very small hole in the skull, and you implant electrodes. That is one sentence that takes about 10 hours, a lot of hours of work to kind of get this thing set up. Then you can record for as long as you can. Normally it's 1 to 3 hours. And then the animal is sacrificed, and then the brain kind of frozen in a certain state, sliced up so that you can look at kind of where you recorded and also what was the state of the network that you were recording from. And again, these silicone electrode probes can get out the local field potential, so that's the kind of the gross electrical activity around the neurons, as well as the spiking activity. And so we're going to look at four things. And I'll go through each of them a little bit, but I don't want to get too technical. Is we're going to look at the spectra, so the actual frequency and power of the global oscillation at a given time. And so again, this LFP data is like kind of the gross electrical activity. And what we can do, this is similar to the picture I've already shown you, is you can go through and say for each time window, are you in a slow oscillation or a theta oscillation? So here on the left is the medial entorhinal cortex, which is near the hippocampus, just behind it, and then the CA1, which is in the hippocampus. So we record from these two regions simultaneously, and you can see this is an example of the electrical fields during slow oscillations, and then faster ones during theta oscillations. And this is analogous, but not the same as non-REM and REM sleep when the animal is naturally sleeping. You can see that there's kind of similarities between these two plots, but this one it's much messier. And what you can do is determine, am I in slow or am I in theta, and then kind of mark it with a color. So here I'm in theta for a very long time, and then slow oscillations, and then theta, and then slow. So I've gone from that very busy, difficult spectral data to this very simple, am I in this or this? And in anesthesia, you can do this. In natural sleep, it's a bit tougher. But now we're going to look at actually what the neurons themselves are doing, the action potentials. And to do this, the first thing we're going to look at is just the firing. So when do things spike? And to do this, we use a sliding window. So we take 10 seconds, we check out what all the neurons are doing that I can see, and then I slide that window 1 second forward and I check it out again. And then I keep going, keep going, keep going, and then you kind of have the firing as a function of time. And it's important to note for all this analysis, we bin. So basically, you take a very small time window, here 20 milliseconds. If a neuron fired, you give that little bin a 1, and if it didn't, you get a 0. So now we've taken all the noisy biological data and transformed it into zeros and ones. We sacrificed a little bit of information that way, but I don't think it's too bad. So first we want to look at the firing. What are the neurons doing? Because what could happen is that the firing rate of the population could be staying the same, but who is firing to create that average could change, and that's important to know. So here's a really cool example. This is one of the reasons this whole work started, is here on the y-axis, these are 67 neurons in the medial entorhinal cortex. And then here at the x-axis is time, and the scale bar here is 1 minute. So every time you see a little blue tick, that is when a spike happened. Excuse me. So you can see there's some neurons that spike a lot, some not so much. But what's really interesting is you can see that there's kind of a group of them fire, and then all of a sudden a different group fires. And then it kind of switched back. And then it did something weird here, and then a different group again. And you can see it's kind of like swapping back and forth, which is really interesting. And traditionally, neuroscientists might say, well, those are different global states. Maybe here you're switching into theta, this is theta, and this is slow, or something like that. Um, but actually, when you overlay the spectral activity, it's not that at all. In fact, during one very long, stable period of slow oscillations, you have very different types of firing going on back and forth. And what we do is you can go through and try to correlate how correlated are the neurons at time A to the neurons firing at time B in these time windows. And you're going to get something out like this cartoon here, where here this is time and time. So red in this cartoon means very correlated. So there'll be times where the population is firing in a very similar pattern, times where it's not, and kind of on and so forth. And if you apply this to the data, this is kind of what happens. You can find this picture here. So this is real data. So this is the picture of this cartoon for this data. Whoops. So you can see there's periods where the firing is very similar, these red blocks. So time, like 10 to 20 was very similar to time 20 to 30. It's really interesting. You have these blocks kind of reoccurring and appearing, and if you go through, you can actually cluster these using an algorithm. I'm not going to talk about that today. And you can kind of mark these with different colors. So you see the yellow type firing is when a certain subset of neurons are very active, and then later you can see they're active again, and then here, and then here. All the red periods are, have a different subset, and the green periods are different again. And just like we did for spectral activity, you can say, okay, if there's 4 or 5 different kind of firing archetypes that we see, yellow, green, red, and some others, we can go through time and mark them down just like we did. And what's interesting, and this is counterintuitive to a lot of typical neuroscience research, is even though we're in a stable oscillatory state, you have this switching of activity, of firing back and forth, back and forth. So here, this is firing like A, B, C, B, D. And again, the colors don't represent intensities, it's just different. And what's interesting is sometimes like this light blue period can occur in both theta and slow periods, but sometimes, like this orange one, it only appears in theta. So substates, these substates of activity are not entirely specific to the global oscillations that are going on. And then we're going to do this with our information theory measures as well. So what I just showed you was just for firing. So information storage, and I'm not going to get into the math today, but it is looking at the mutual information of a neuron's spike train, these zeros and ones, from one time bin to the next. So you take 10 seconds of zeros and ones, you shift it 1 second, and you have another 10 seconds of zeros and ones. And you say, how much information do I have about time B given that I know time A? Or you could say it opposite. How much do I know about A given that I know B? Or how uncertain am I about what's going to happen next based on what I know now? And this is a measure taken from Claude Shannon's work. It's similar to an autocorrelation function, if you know those, but it's nonlinear. So you're asking, and I can send articles to whoever wants. You can contact me about kind of the establishment of this measure of active information storage. So there's kind of information being actively stored in the temporal patterns of neuronal firing. And what's interesting is we can do the same process as we did for firing, but for storage. And this is the same recording as the one I just showed you a second ago. And you see that this picture looks radically different than the one I showed you a second ago. You see these blocks of activity, and they change through time. And again, you can cluster them. And what's really interesting is there will be moments where the firing rate stays the same, 5 hertz to roughly 5 hertz, 2 hertz to 2 hertz. But the storage going on is very different. This is kind of easily captured here. So this is the correlation patterns for firing. Here's the correlation patterns for storage. You can see there's some similarity, like this big block here. You can almost see like a hint of it here. But the storage that's going on is very different than just the firing patterns going on. And again, you can cluster these just like we did for firing. Some of them occur in slow, some of them occur in theta, some are kind of mixed, and we can mark them. And again, they're not entirely state specific. Then finally, we're going to look at this information sharing. So how does information propagate around? It's the same measure, mutual information, but instead of one neuron to itself, we're saying, if I know the firing pattern of neuron A, how much do I know about the firing pattern of neuron B or C or D, blah, 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 all the way to the end. And again, so I just saw Santosh ask, how is storage defined? And this works for this. So storage is the mutual information of a neuron's spike train in one window and the subsequent window. So you take the spike train, it's binarized. So you have 10 seconds of zeros and ones. You shift it one, and then you look at it again. And you say, how much information do I have from here to here? The mutual information between these two trains. Sharing is the exact same thing, but instead of one neuron to itself, it's one neuron to any other neuron in the network. So now we're asking, instead of just the temporal patterns of neuronal firing, we're looking at the spatiotemporal patterns of firing. So how are these patterns propagating across the space of the network? It's, again, similar to a correlation function, but it's nonlinear. And this graph looks a little bit different, but you can see that, again, there's this blocking pattern that arises. Again, it's not global state specific, and you can mark when they change using the color scheme we used before. So now we have this nice big picture of the dynamics of the network through time, the spectral activity, the firing activity, the storage activity, and the sharing activity. And again, they're not all one-to-one. Sometimes they change altogether. Like this is pretty close here where I'm highlighting with my mouse. But most of the time they seem somewhat different and independent. And what's interesting is what we did is you can go through and label each state with a symbol. So like I talked about earlier, like B, D, C. And so you can talk about your recording as a sequence of symbols through time. It's like a word through time. BBB can be thought as a group of symbols or a word. And so now you have this kind of language that is expressed through time. So when I want to describe the dynamics, we can then kind of lean on language tools to talk about, what's the language these things express. I'm not saying this is a literal language. It is just a clever way to analyze the dynamics. So to kind of wrap that up, we observe these switching substates of firing, storage, and sharing, even though we're in stable periods of global oscillations. And what's interesting is that the substates are not entirely determined by global states, and the transitions don't seem to be deterministic either. And we're going to use this kind of representation to talk about kind of how the information processing states, the states being this kind of vertical slice through time. Boom, information processing substates. So that's what we observed. I'm going to skip through this part because I want to get to something else. I'm happy to talk about it later if someone wants, but the sharing networks are really buck wild. But I want to make sure that I get to some other stuff first. And to talk about, again, we wanted to talk about the organization of this. I mentioned that we can use these cute language tools to analyze the organization of these dynamics. Again, I'm not saying the brain has this is the language the brain speaks, but you can put these words through time as a sentence and analyze them like you would a traditional language to talk about how they are organized. And again, if you remember back to the beginning in my introduction, the two extremes I gave were totally regular, like a library, totally random, like a destroyed Amazon warehouse, or something in between, like my old boss's office. We can use that same idea of comparing the tables that we see between the bounds of regular and randomness by computing the complexity. And this is similar to the Limpel-Zeev complexity, for those of you who are familiar with it. Essentially, you measure how hard is it to reproduce this table with a piece of computer code. This one you can imagine is very easy to reproduce. Print A 4 times, B 4 times, C 4 times, D 4 times, and I'm done. It's very easy to compress this because of the structure. The random one would take a very long piece of code to write because essentially you have to print every single line. Print C, B, C, C. Da, 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 da. There is no structure, therefore no compression, and the code it takes to reproduce it is very long. The complex one is something in between. There are parts that you can compress and parts that you can't, and these algorithms can measure how complex something is. And what's interesting in this first paper, we'll just look at the graph here on the left, is that during periods of theta, the complexity was higher than in periods of slow. And it was well above 0. So no matter what, if we looked in anesthesia, in natural sleep, or across different regions, it seemed that theta, maybe because the oscillations are faster, allowing for words to change quicker, was more complex than slow. And so we kind of established that there is a complex organization of information processing going on, and it seems to be modulated by oscillatory state, but not entirely determined by it. So that was kind of the end result of Project One, and that's published in Science Advances, called Computing Hubs in the Hippocampus and Cortex. I didn't talk about the hubs. They are very cool. I just wanted to focus on the organization side of things. But now, kind of the cool part, what I'm working now and what I'm working towards in the future is how this all changes in disease and how does disease models help us understand what's going on normally. That's very neuroscience to do. This is how any neuroscientist knows anything is we watch what's happening normally, or we observe what's happening normally, and break something and see how it changes. Like the only reason we know the hippocampus does what it does is because someone lost theirs, right? HM in a surgery, and all of a sudden he couldn't form memories, and we were like, well, I guess that's important for memories. And there's been a series of things like that throughout neuroscience history that kind of gives us an understanding about what parts of the brain do. But essentially, neuroscientists will observe things normally, we will break it, and then hopefully in the breaking figure out maybe parts of how it worked. Uh, it's not the best way, but it's what we've got. And again, all the experiments that I do in epilepsy are very similar to the ones that I just showed you. So we take rats, they are put in under anesthesia, and we record for a certain amount of time, then they're sacrificed and the brains are analyzed. That's called acute recordings because you do it one time. Chronic implants mean you can record for many, many days, maybe as the animal is learning something, etc., etc. I do that some too, but these experiments are all acute. Now, the important difference is all of these rats had epilepsy. This is a model of temporal lobe epilepsy. It's a really common form of epilepsy. And what you do is you inject chemicals into them. You wait for like an hour. They have a massive, massive seizure. It's this huge bit of trauma. You give them some more drugs to go to sleep. And then after a couple of weeks, they will have spontaneous recurring seizures. And a lot of the phenotypes of change inside of the neural networks inside of their brains are very similar to the ones you'd see in humans. So that's what you do. You have rats, you cry, and then you make them epileptic, and then you cry about it, and then you analyze their brains afterwards. It is not a fun procedure. And so what we wanted to do is analyze, again, these information processing states. But the question we had going into this is like, one, are states even there still? And two, how many should there be? I very cleverly didn't speak about that earlier. And so what we did is instead of using some kind of very clever algorithm to decide how many states I think there should be, what I did was did the same thing. I built my similarity matrices, the ones with the blocks that I showed. Hold on, my dog is barking. Hazel. She's very upset that someone's outside and she's not. And so what we do is we take those similarity matrices that I showed you with the blocks, and then I cluster them a number of different ways. Here in this example, what if there are 3 clusters? It would look like this. 4 clusters, 5, 6, so on and so forth, up to 10. Someone asked, any suggestions on a good tutorial for learning brain terminology? Oof, That's a really good one. There's some textbooks out there that are really good. I think I have my email at the end, and you can email me, and I'll send you some stuff. Um, there's a really great book by Georgi Bujaki. That is a very hard name to spell, but he writes some incredible books. It depends if you're interested more on the theoretical end, or the computational end, or the biological end. So feel free to comment, and we can even chat in the chat after this about some, because each path kind of has a little bit different terminology that changes depending on how you view things. So anyway, they, instead of clustering it just one time like I did in the past and made those nice pictures that I showed you, I cluster it many different things, many different ways, 3, 4, 5, 6, 7, all the way up to 10. And the first thing we did is we wanted to establish that the clusters were actually doing different things. So like, what makes one state different from another one? I showed you the thing for firing where it's just like, oh, these neurons are firing and then these neurons are firing, and it's different neurons, so they're probably doing something different. And so what we did is built this measure called contrast, where you look at the activity of a neuron in a substate, like the green one, for example, and then you look at its mean activity throughout the whole recording, and you subtract those two things and then divide it by the mean. So what that's going to look like is that if the behavior inside of a substate is roughly the same as the behavior across the whole recording, this measure will be 0. If the behavior in the substate is way more than you would expect to see on average, this bar will be very high, and then the opposite. If it's way less, like for example, if the behavior is 0 but on average it did 10, this number will be very negative. And so what you see here in the pictures are these bars. Each bar is a neuron, and then how far up or down it goes is its contrast in a given state. So here in this one, most of the neurons were firing less than they normally did, except for this little dude. In this dark blue state, it's like half and half-ish. And the light green, you see a lot of them are firing a lot. And then in the dark green, a lot of them are firing a lot, but it's a bit different. Notice how this one was firing a ton, and then boom, it's all the way down here. So you're seeing like little snapshots of what's happening inside of these states. And you can do the same thing for storage. So each state, I mean, visually it looks different, but you can quantify how different. I'm not going to go into that, but you can see that it's different. And then for sharing, it's different as well. So the brain seems to be organizing some of its behaviors into these substates, and what's just absolutely buck wild is despite all the damage that's done in epilepsy, these states still exist. And what we wanted to look at is compare the contrast, the average contrast that we see in control and epilepsy. So, okay, if both are organized into states, how different are the contrast values in control and epilepsy? Buck wild is a great term, and I use it in all my science talks, so I'm glad someone out there appreciates it. So what we did here is, let's say we take firing, for example. I'm going to look at the mean contrast value from a bunch of my recordings in control, and I'm going to randomly sample like 80% of it and find the mean contrast that I find. And then I'm going to do the same thing in epilepsy, and I'm going to subtract those two things. So the mean of control and the mean of epilepsy, I'm going to subtract them. So if it's positive, control was more contrasted. If it's negative, epilepsy was more contrasted. And if I do that like 5,000 times, because I'm randomly sampling, I get this nice distribution of the difference of the means. And so what we see here is that for the control case, the contrast is positive, meaning that if you were to look at this picture from all the control files, all the bars would be way bigger for firing than you would see in epilepsy. So the behavior is kind of varies about the mean quite a bit more in control than they do epilepsy. Interesting. However, for our information processing measures, epilepsy seems to be way more contrasted. So despite not having as much contrast in their firing, the information processing is way more contrasted. So there's kind of a shift in the internal state behavior. And what's really interesting is, I was going to show it, but it doesn't depend on how I cut it. This always stays the same. And we also did a lot of other tests, like for example, testing how well the data is clustered. And I have a bunch of nice, basically supplementary figures. And I'm pretty happy to conclude that that states still exist in epilepsy, um, and that their constituent activity sometimes is more variable than what you would expect to see. We also looked at the state specificity. So again, if you remember, in the first part of the talk, some of the states only occurred in slow oscillations, some of them only occurred in theta, and some of them were like at a nice in between. So again, I always like to think about the two extremes of this. So this type of measurement you can capture by taking the probability of finding a state in theta minus the probability of a state occurring in slow. So basically, if it's 0, that means these things are the same. So it's going to look kind of like this. So most of the time you see your state in both, halfway between theta and halfway between slow. If this value is 1, that means it either only occurs in theta or only occurs in slow. And so it would look like this, more of this U shape here. So 0 is kind of this peaked in the middle, and then 1 is kind of this U shape thing. And what we did is found that when in epilepsy, in the case of firing and storage, they lose this specificity. They go from this U shape back to this middle state. It's like all of a sudden you're mixing the nonfiction and fiction books more than you should have. Sharing doesn't do this, and I don't know why, but it doesn't. Yes, Santosh, it is interesting that epilepsy is characterized by more global firing. How I'm kind of interpreting it is that if there's more synchronous firing, the contrast would be less, right? Because they're kind of locked into a type of firing for long periods of time. So the global mean better represents what they're doing inside of the substate. But then when you look at the storage and the sharing, the actual where the firing is happening apparently is a bit more erratic and maybe giving rise to way more storage and sharing, more than you would expect to see. So the contrast would be higher. If that's how I'm currently thinking about it, but I could be interpreting it wrong. Um, it's a work in progress. Um, so states still exist, and the relationship between substates and oscillatory state is altered. Now, back to the big question. Again, library, super regular. Destroyed Amazon warehouse, very random. Complex is something in between. What does epilepsy look like? So what I did is I took all the files that I had, all the different ways I could cluster this thing, and ordered them as much as possible. So like this is as much of a library as I could get. This is as ordered as I could possibly make it. Then I take a table and I shuffle it in a lot of different ways to make it as random as I can. And then I have the one that I recorded. So now anytime that I measure the complexity of something, I can say, is it closer to this one or is it closer to this one? And I didn't mention it before, but there's another aspect of complexity, and that's the dictionary. So if you have a certain set of symbols, there's only so many different ways you can combine them, and that's called the dictionary. So if I have if I have 3 different symbols and they can take 3 different values, there's only so many different ways I can put those together. So a low dictionary would look something like this, where you only express a couple of the possibilities. A high dictionary would be like you express almost all of them. And so what I did is I measured the dictionary for all the things that I found. Then I measured the dictionary for the most ordered tables. So I ordered them as much as I could. And then all the tables that are most random. And we're going to ignore the x-axis for now, but just look at where the blue and the red curves lie. So in epilepsy, red, this dictionary goes up in both cases, in both regions of the brain that I'm recording from. So what's interesting is all of a sudden in epilepsy, you're combining states together in new ways or in more ways than control did. Maybe that's not such a great thing. Maybe some of those combinations were dangerous or not meant to be. You're delving into unknown space. I'm not quite sure. It's just that this measure goes up and observable. But dictionary plays into complexity, right? If you have more words that you express, the complexity could probably go up. Um, yeah, I don't know why I have that slide twice. Sorry. And that's what we see. So here is the measure of complexity, and this is actually, I've switched measures, so this is actually the Lempels-Eve complexity, if you're familiar with it. And again, 0 is, I normalize this measure such that the most ordered tables represent 0, the most random tables represent 1. And then I measure the complexity of the tables that I find in control in epilepsy. And again, for now, ignore the x-axis because I just wanted to get the general message across that in epilepsy, complexity goes up. So essentially what we're finding is that what's interesting is the control is actually sitting quite low relative to as ordered as possible. It's complex, but it's not like buck wild. And in epilepsy, that complexity goes up. So you're looking closer to the Amazon library or to the destroyed Amazon warehouse than before, which is, I guess, bad because epilepsy is bad. But it's not like huge, right? So the states still exist. They are more complex, but it's not like it's shooting up into randomness. There is still some amount of order. It's just shuffled around a bit. They lose the organization from the theta and slow. They make some more states than they should have, maybe. So that how the information dynamics in epilepsy has become disordered. And that's kind of the slide characterizing that. So overall, we find that the IPS organization is more random. And that's published in bioRxiv under this name. It's almost published. It's a nightmare. It's almost done. So going over the big question was, is organization related to collective neurocomputation? So again, I wanted to look at state-dependent information processing in this switching. So I think we found information processing states, so a hint of this. And the non-trivial state switching, our tables were complex. So if they had been trivial state switches, the complexity would have been much lower. And while we can't know what the brain is computing, we could still search for signs of that organization at an algorithmic level, one that I think is sorely underinvestigated. And then the big question was, what does pathology reveal about organization? Is that in epilepsy, and I believe you might find this in other diseases as well, this complexity goes up. Something about the algorithms, this kind of instruction set, gets messed up. So yeah, the bottom stuff is messed up, but then now the level above it, these instructions are changing. It's like imagine if you were going to build a castle, a Lego castle, which I love to do. You have blocks, a certain set of blocks, a limited number of blocks, and you have an instruction set. But what if I was a huge jerk and I went through and filed some of the blocks into different sizes, into different shapes? I twisted some under a torch a little bit. I messed up, I don't know, half of your blocks, 80% of your blocks. If you tried to put it together following the same instructions, it wouldn't work so well. You might be able to achieve it. The castle is still technically somewhat of a castle, but it's less structurally sound. In the same way that in epilepsy, um, you're still alive, thank God. Sometimes it can result in death, and that is awful, but most of the time, people continue to live. It's still a castle. Um, but when we think about treatment plans, and this is what I'm investigating now at Mike's lab, does it make sense to really go back and try to fix every single little block that you can find in your castle? I mean, that's a lot of pieces. Rather, could you build a new instruction set? Maybe we could intervene at the level of algorithms to build a more stable, better castle, given the fact that our pieces are different. And so that's kind of what I'm doing in Mike's lab now. Keep an eye on that space. Um, do organisms reuse algorithms in different spaces? Can relationships between algorithms and certain phenotypic behaviors be established? Can you manipulate algorithms? Can you translate algorithms? Can I take the algorithms from one organism and use them to do something in a different organism? How do I get two organisms using two different algorithms to understand one another? I don't know. And what happens when you take an artificial body and link it with algorithms or behavior? What happens if I let neurons control a robot and interpret these algorithms a certain way, and then put that into feedback loops? How does that change the algorithm? Can they change themselves, or are algorithms hard-coded and immutable? I don't know. So thanks, Proteus Institute. This was fun. It's strange not being on Zoom, but I've been seeing chat. It's great. Here are the people that funded me during this work. Now I'm at Mike's lab, but before I was in France and was paid by the Marie Curie group or Project IMGATE, by INSERM, and then the European Commission. So thanks, Proteus Institute. I mean, we have like 8 minutes left. I sped through that, I guess. Sorry. So if anyone has any questions, put them in chat. I'll hang out until 3. Yeah. So even if you have questions unrelated to what I just presented, feel free to type them in the chat. I'll paste some links for people. Now. This is for Faten Nubus Bis. I don't know. This guy has a book called The Brain from the Inside Out. It's very good. It's kind of the updated version of an older book that he had called Rhythms of the Brain. Also very good. If you can't find the kind of Inside Out, they're just kind of roughly the same book. He just updated a lot of the ideas and rewrote it. The Inside Out is a little bit nicer overall. Do you think that the word repertoire would be different post-epilepsy compared to pre? Yeah, so that's what I was trying to do when I was in my PhD, and unfortunately, COVID struck and all experiments had to be stopped. So I didn't get to finish, but some people are doing the experiments now, and hopefully we can apply all this stuff to it. Just I didn't get to do the experiments. So I think the essence of Santash's question is, if you have an animal and you're recording from it, let's say you can establish these states in natural sleep, which we've shown you can do, and then you make it epileptic and you put it, you let it sleep again. How does everything shift? Can you That's a super cool question, and I hope that we can answer it. There is an enormous amount of technical difficulty hidden under that. So to chronically implant a rat, you have to put it under anesthesia, which is fine. Implant the probes, secure it to the skull, and then basically give it a little protective hat so that it doesn't mess with the electronics. That's how all chronic implants are done, and they're pretty happy with them on their heads. They're not too heavy. That's fine. You can record for a long amount of time. Unfortunately, the brain doesn't like plastic in the head. So eventually, a scar tissue will start to form and you'll lose signal. So there can't be a lot of, you can't let it last for like 6 months or a year I think people are working on ways to get them to last longer. But then the worst issue is that when you make an animal epileptic, in this case yeah, I'm glad you enjoyed it. So when you make the animal epileptic, there's this large staticus epilepticus. So they have these massive seizures that causes late stage epilepsy, so the spontaneous seizures. And it's like the seizures are a nightmare. Like they rip and thrash around their cage. And you can imagine if you have very sensitive electronics, one, they might bust, or two, you essentially have these probes are sometimes like two shanks. You essentially have like a fork in Jello, and if you get unlucky, when the brain moves, if they were to hit their head, um, the fork kind of like you can essentially kill it. So having an animal live through that is just really tough, and I actually don't think ethically people can do that. Um, you would have to be able to prove that you could do that without having the animal suffer, which I don't think you can do. I would never, yeah, exactly. I would never want to be a reborn as a rat. We do treat our rats really wonderfully. It's something I take a lot of great pride in. We've developed a lot of protocols. Someone trained me, and I always redirect them back to her. She's a wonderful scientist in the Netherlands. To habituate the animals, they live in groups, and you try to they're like small puppies. So this whole procedure just is awful. But and it can drastically also affect the science if you don't treat them well. all your data is basically wrong for a number of reasons. But yeah, you try to. So I think the word repertoire would be different, Santosh, but to experimentally do this is kind of a nightmare on a number of levels. Also, ethically, it's really bad. So what I'm planning to do is, what if we can grow a neural culture? So this is like you take neurons from a brain. You either can grow them from stem cells, or you can extract them from the brain and grow them on a dish. You can record them for a little while. Then you could put some drugs on there to make the neurons alter a little bit to be more like it would be in epilepsy. So again, this is now no longer in vivo, right? It's in a dish, but you don't have to worry about ethics now. It's just neurons in a dish. Well, I hope. I mean, I don't know. Maybe they are sentient and we're awful. But essentially, if you have cultured neurons before and then you make them epileptic and after, you could apply the same principles. And I think, yeah, I think you would see more words. The repertoire would be different and the complexity would go up. It would be wonderful to be able to track that transition from control into epilepsy or into any other disease, right? I mentioned this as well. I think you would see a lot of these algorithmic changes across diseases. And one of my hypotheses is that by learning to intervene at this algorithmic level, you might be able to better treat disease in a number of ways. Yeah. Any other questions? It's hard because I don't see anyone's face because we're not on Zoom. Let me pull up the stream to see if people are still here. Oh, yeah, there's still people here. So. But thanks for being here. I appreciate it. I'll put my email in the chat if anyone wants to email me questions. I'm happy. I should have put it on the slide, but I didn't. I'm happy to chat with anyone. I'm also on Twitter. I don't use it all the time, but I'm there. So I think with that, I'm going to wrap it up. I hope everyone had a good time. This was recorded. I will send it to the appropriate people, and I think it will be uploaded on YouTube soon, if for whatever reason you would like to rewatch this. Okay, I'm going to try to turn off the stream now. Wish me luck. Stop streaming.